Greetings, everyone, and welcome to another Genetic Engineering and Biotechnology News webinar series. Our presentation today is entitled Streamlining Glycan Analysis, from Sample Preparation to Data Analysis. I'm Jeff Bogaliskas, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. Glycosylation has had a tremendous impact on the biopharmaceutical industry as a fundamental attribute to monitor during the development and production of biotherapeutic proteins, and regulatory agencies now requiring in-depth characterization using state-of-the-art analytical methodologies. Investigators are constantly on the hunt for glycan preparation and analysis tools that will save them precious time and money. Let's meet our speaker for this GEN webinar, who will describe a streamlined glycan analysis platform that begins with sample preparation and is followed by data collection and processing, which includes the incorporation of a glycan library search tool for accurate structural identification. Bill Alley is a senior scientist for the Waters Corporation. Dr. Alley will describe methodology for sensitive glycan measurements that take advantage of a simplified high-throughput sample prep procedure utilizing the GlycoWorks RapiFluor-MS N-Glycan Kit. Before we get rolling into Bill's presentation, I want to encourage everyone to submit questions for our Q&A session at the end of the presentation. We'll try to answer as many questions as we can, so simply type your question into the Q&A box on the left-hand side of your screen and hit submit. All right, with all the particulars out of the way, let's get our webinar started. Bill, the audience is listening.
Hello, everyone. I'd like to thank you for attending today's webinar entitled Streamlining Glycan Analysis, from Sample Preparation to Data Analysis. I'd like to start off today's talk with an outline of what we'll be going over. We'll first discuss glycosylation basics, why glycosylation is important, and why we want to study it in the biopharmaceutical industry. I'll then discuss RapiFluor-MS, a molecule that we developed at Waters that's been available for about a year and a half now. And RapiFluor-MS really helps out with both FLR and MS detection. I'll then discuss how we integrate RapiFluor-MS into an automated analytical workflow that includes a library search. The library search really helps out with being able to assign structures and compositions to our FLR peaks. We'll then conclude by presenting a real-world example where we're comparing Innovator and Biosimilar Infliximab using this analytical workflow. I'd like to start off by just showing the basic building blocks, the basic monosaccharides that are used to construct N-linked glycans. And in today's webinar, we'll be discussing only N-linked glycans. I'd like to point out a few of the important monosaccharides, one of those being the fucose down in the lower left-hand corner, and the N-glycosylated acid over on the lower right-hand corner. And glycosylated acid is a non-human monosaccharide, and so its presence on biopharmaceutical products can cause some difficulties if present in the human body. We also have galactose, which when linked via an alpha linkage with another galactose is also non-human and can cause some problems if present on a biopharmaceutical product as well. There are three main classes of N-linked glycans. There's the high mannose class, the complex class, and the hybrid class. N-linked glycans are composed of a common chitobiose core, which is composed of 2 glycnacs and 3 mannose residues. The high mannose class of glycans extends the chitobiose core with only mannose monosaccharides. The complex class of glycans will extend the core with a glycnac, which can then be further extended with a galactose and further extended with a sialic acid. Complex glycans can also be fucosylated on the core or on an outer arm. Complex glycans can also have what we call a bisecting glycnac, which is attached to the core of the complex glycan. Hybrid glycans are a combination of the complex and the high mannose types of glycans. Glycosylation is an important modification of proteins. And if we think about the human body, at least 50% of proteins are glycosylated, and glycosylation can influence a number of properties of the particular protein. So it's not surprising that a number of biotherapeutics are also glycosylated. Glycans can be attached enzymatically to a protein through asparagine residues, and we call these N-linked glycans, or they can be attached to serine or threonine residues, and these are called O-linked glycans. In this webinar, we're only going to focus on N-linked glycans. N-glycosylation of proteins is routinely characterized and monitored during the development and manufacturing of biopharmaceuticals. And as we've just discussed, glycosylation influences a number of physicochemical properties of biotherapeutic proteins. If we consider a MAB that we see on this slide, the levels of core fucosylation can modulate its ADCC, and its levels of high mannose glycans can alter its serum half-life. Sialylation is interesting because it can lead to anti- or pro-inflammatory responses, and non-human groups like N-glycolyl neurominic acid or 2-galactoses connective via an alpha linkage can induce immunogenic responses. So with all of these different properties that can be influenced by glycosylation, the glycan profile of a biopharmaceutical is important to monitor, and it's also a critical quality attribute, and glycosylation must be defined as rigorously as possible. This slide further highlights the importance of glycosylation for biopharmaceutical products. Again, just giving some more specific examples where sialylation can decrease the serum half-life of EPO from 2 hours down to 10 minutes. Glycosylation analysis can be a very challenging task for multiple reasons. Unlike proteins, glycosylation is not template-driven, so for a single composition, that is the collection of monosaccharides that make up a given glycan, it's possible to have several different structures, or how the monosaccharides are connected together. For example, many glycans contain a fucose, as we saw a couple slides ago, but fucose can be located on the core or an outer arm. Sialic acids can also be connected by an alpha-2,3 or an alpha-2,6 linkage most commonly. And unlike proteins and peptides, glycans aren't linear chains. They're very highly branched structures. And adding to the complexity, glycosylation is also very sensitive to environmental or biological changes. So slight changes in our cell culture conditions or our cell media can result in significantly different glycan profiles. Analytically, glycans can be quite challenging to analyze. They're not really ideal analytes for LC-MS. They don't have an appropriate chromophore for detection by optical methods like UV or fluorescence. They don't have a basic site for ionization in the positive mode of mass spectrometry. Therefore, glycans require extensive sample preparation and derivatization so they can be successfully analyzed by LC-MS. However, many sample prep protocols are very time-consuming. They can range from anywhere from 4 to 5 hours out to several days, and they can be very tedious and require significant skill to be able to get reproducible results. And on top of that, we haven't even talked about analyzing glycan LC-MS data. While many bioinformatic tools are widely available to automate protein analysis and peptide mapping experiments, these tools aren't as widely available for glycan analysis as we'd like them to be. So many scientists still have to examine their data and make structural assignments by hand. And there can still be some ambiguities in this first pass, so a lot of times you have to go back and conduct tandem MS experiments. And interpreting this data can be a bit tricky as well. So to address these analytical challenges and fill in some of these gaps, Waters has made significant investments in the area of glycan analysis, both in sample preparation and in developing bioinformatic tools. As we discussed on the previous slide, glycans are very challenging molecules to analyze. And again, analytically, they're not the ideal analytes for optical or mass spec methods. Some glycans contain sialic acids, and they can be detected in the negative mode of ionization, but it's more difficult to study neutral glycans in this way. So this isn't really a universal method. So to be able to detect all the glycans, they do need to be derivatized. And in general, the derivatization methods that we have developed, we can consider them being grouped into different types of buckets. So we have one bucket that can enhance optical detection, one bucket that can enhance mass spec performance, and I've included a third bucket for quantitation by mass spec since they have their own unique molecules. If we look at the optical bucket, we see molecules like 2-aminobenzamide or 2-AB, 2-anthranilic acid or 2-AA that can help out with both UV and fluorescence detection. Another molecule that's widely used, particularly in capillary electrophoresis with laser-induced fluorescence detection, is 8-aminopyrene 1,3,6-trisulfonic acid, or just APTS for short. If we look at some of the things that can enhance mass spec performance, we see permethylation, which has been widely used, or hydrazide tags that can increase the glycan's hydrophobicity, or it can impart some more basic groups to enhance the mass spec ionization efficiency. But overall, we can see that there's not much overlap between the two buckets for optical and mass spec performance, with the exception of procanamide. It does help out with both optical and mass spec performance. So this lack of a single molecule that can enhance both mass spec and optical detection and be able to label glycans rapidly was really an area that we wanted to invest some time and effort into to be able to develop a molecule that would be able to do all of that in a single step. The molecule that we developed that can help out with both fluorescence and mass spec detection, along with labeling glycans in a rapid manner, we call RapiFluor-MS. And RFMS takes advantage of our long history in rapid labeling of amino acids using our AccuFluor tag. And AccuFluor is shown in the upper right-hand corner, and we can see that it has a rapid labeling group. That's the NHS carbamate, and it has a quinolanil group that allows it to be very active in the fluorescence detection. And if we take a look at RapiFluor at the bottom of the slide, we can see that RapiFluor is really a second generation of this molecule, where we've gone in and we've added on a tertiary group that has a nice gas phase basicity, and it can pick up protons and really help out with the ionization efficiency of glycans. One important thing to remember when using RFMS is that we are labeling glycosylamines. We don't allow the glycans, once they've been released, to be converted back to a reducing sugar with the hydroxyl groups. This type of chemistry doesn't work well for those types of molecules. But by working with glycosylamines, we're able to take advantage of the rapid reaction kinetics of the NHS carbamate group. The labeling of the released N-linked glycans is done in an almost instant manner, in just a few seconds, although we recommend letting the reaction go for about 5 minutes. Following the reaction, we'll see a shift of approximately 311.16 Daltons. RapiFluor-MS labels glycosylamines almost instantly. While the time investment in the labeling step has been minimized, the upstream sample prep workflow is still very time-consuming, and it can still be very labor-intensive. So it's not uncommon to spend several hours, up to several days, preparing the glycans just to get to the labeling step. And then the labeling, again, it can take several hours plus the cleanup. So it's very important for us to ensure that the sample prep workflow prior to the labeling could be done on a much faster time scale to take advantage of the rapid labeling of RFMS. So we set out to redesign the workflow, and we invested quite a bit of time in being able to do this. And I'd like to discuss that with you now. So we begin with 50 micrograms of a glycoprotein. It can be a monoclonal antibody, some other biotherapeutic, but it also works just as well with any other glycoprotein. We denature it at 90 degrees C with 1% RapiJest. That's a little bit higher than what we use for peptide mapping experiments, and we do that for 5 minutes. Then we let the sample cool down to room temperature, and then we add rapid PNGSF that we co-market with New England Biolabs to release the N-linked glycans. We can get complete deglycosylation in 5 minutes at 50 to 55 degrees Celsius. The fast deglycosylation is very important to help preserve the glycosylamine and not allow it to be converted back to reducing end. Then we label the released glycans with RFMS, and we recommend a 5-minute reaction. And then we do need to purify the glycans to remove excess labeling reagent. And we use a microhelic elution plate, and this takes about 10 minutes. So depending on the number of samples, the entire sample prep workflow from beginning to end can be accomplished in as little as 30 minutes. Although if you're going to be preparing 48 or say 96 samples, the time investment in sample prep will be a bit longer, but it's not going to be too bad. Following the sample cleanup, we just dilute the samples down with some acetonitrile and dimethylformamide. We don't dry them down in a speed bag, which also saves time. And then once we've diluted the samples down, we can just go straight into the UPLC and inject and do the mass spec analysis. With this new workflow, we do have a robust method that gives us high yield. We're able to achieve complete deglycosylation in only 5 minutes. We've verified this through a number of different tests, including intact mass analysis and gel shift assays. For monoclonal antibodies, we've looked at subunit by LCMS. We also have a very efficient labeling procedure. We can get yields of at least 95%. And we do take a bit of a hit on the solid phase extraction, but we're getting a total recovery in the area of 74 to 75%. But for solid phase extraction, I don't think this is really too bad. I think it's quite good. I've done SPE in the past and had a lot more sample loss, so I'm quite pleased with the 74%. So overall, we're getting a total yield of about 73 to 75%. So that's actually really good. The derivatized glycans are quite stable, as this slide shows. On the left, we're comparing the FLR traces of a sample that was subjected to UPLC-MS analysis immediately following the sample prep. And on the bottom, on the left-hand side, we're looking at that same sample that was analyzed 3 days later. And these traces are virtually identical. On the right-hand side is a chart that's showing the FLR peak areas for several monoclonal antibody glycans. And we can see that there are no changes in the peak areas after 3 days. So we can guarantee that the derivatized glycans are stable for 3 days, but we can extend this out a little bit further, so maybe up to a week or so before we start to see any noticeable loss. It's really important to have stable derivatives because if we prepare, say, 48 or 96 samples, that can take a little bit of time to get everything analyzed by the UPLC-MS analysis. But having stable derivatives for a week, we're very confident that we won't see any compromised sample integrity over that time period. To benchmark RFMS, we've compared it to several other commonly used tags, including Instant AB, 2AB, and Procainamide. And here we're showing the response factors of the other labels as percentages of the response factor of RFMS. And the gains in fluorescence and mass spec sensitivity are apparent in these two plots, since it does portray the response factors for the other molecules normalized to that of RFMS. On the left, we're comparing the performance of the FLR, and RFMS gives the highest signals, with Instant AB giving the next highest signal, but it's only about half of what we get for RFMS. On the right, we're comparing the mass spec performance, and again, RFMS gives us the highest signals, with Procainamide this time coming in second place. But it's only about a third of what we see for RFMS. This is actually a very interesting result, since both of these labels contain a tertiary amino group. And we believe that the enhanced signal intensity of RFMS labeled glycans originates from RFMS having a bit more hydrophobic character than the Procainamide label. And there are some studies out there that show that additional hydrophobic surface area for a glycan does lead to increased electrospray ionization efficiency. So RFMS is the first molecule on the market that can label glycans in an almost instant manner, improve the fluorescence signal detection, and improve the mass spec performance. Now let's take a look at how we can integrate RFMS labeling into a streamlined analytical workflow that includes searching an RFMS glycan GU scientific library. To make the task of glycan analysis easier, we've developed an application solution that streamlines data collection, data processing, and data analysis. This solution has three main components, the first being the hardware, the second is chemistry, and the third is software. The hardware is our analytical system and includes an Acuity UPLC H-class biosystem, and that has an Acuity UPLC column manager, an Acuity UPLC FLR detector, and a Xevo G2XS QTOF mass spectrometer. The chemistry includes the GlycoWorks RapiFluor-MS N-Glycan Kit and an Acuity UPLC Glycan BEH amide column. The software that we use in this solution is our UNIFI scientific information system. And when we integrate all of the components into a single system, we're able to streamline the process of data collection all the way through to reporting. And this solution makes the task of glycan analysis much easier and faster. Now let's take a look at how UNIFI works, and in particular the glycan application. On the left is a generic workflow within UNIFI. Data acquisition, processing, the bioinformatics, and reporting tools are all integrated and automated within the UNIFI scientific information system. And UNIFI is a compliant-ready architecture. In the middle is our HILIC FLR with accurate mass workflow. This solution allows the routine assignment of N-link glycan structures using HILIC UPLC data from the FLR that has been time-aligned with the mass spec channel. The chromatographic data are then processed and calibrated and converted into glucose units, and we'll talk about glucose units more in just a bit. The mass spec data are similarly processed with the lock mass calibration. Our Hillock FLR with accurate mass data are then searched against one of our GU scientific libraries for structural determination. And this search feature simplifies and expedites the task of structural assignments. UNIFI also offers an alternative workflow for DDA types of analyses. The data are acquired and then processed, and during the processing, the data are deconvoluted and de-isotoped. The file can then be converted and exported as a .lcs or as an MZ XML file. And these files are compatible with the SimGlycan software that can help automate the task of tandem MS structural characterization. And I would just like to point out that the UNIFI glycan workflow does support both 2AB and RapiFluor labeled glycans. It's important to note that the workflow that we recommend is a dual detector method, and we do utilize both the FLR and mass spec channels. We use the FLR for glycan library searching and for quantitation. And our quantitation, typically we use a normalization scheme where the FLR peak area of a particular glycan is expressed as a percentage of the total peak area for all of the glycans identified by the library search. We don't use the MS channel for quantitation. We use that information to confirm our GU scientific library searches. And I'd like to point out that RFMS has greatly helped out with our GU with accurate mass workflow, particularly for the lower abundance glycans. And in this slide, I'm comparing the FLR trace on the top to the mass spec base peak chromatograph and base peak intensity trace on the bottom. And this shows really how closely the two detectors mirror each other. And of particular interest are those glycans that elute later in the run and are present at very low abundances, as we can see in the insets. Using other tags, we really struggle to get high-quality mass spec data to confirm the library search results. But with RFMS, we're able to get enough mass spec signal to be able to really confidently identify what those particular glycans are. And as a general rule, for me, if I see a peak in the FLR with an intensity of around 0.2, I'm quite confident that I'll get good enough mass spec data to get that accurate mass confirmation. Of course, this is a very rough estimate, as all FLR detectors and bulbs are unique as our mass spectrometers. But again, for me, this general rule works out quite well. And so now let's talk about the RFMS GU scientific library in a little bit more detail. This library is very similar to our 2AB library. So if you're comfortable using the 2AB library, using the RFMS library isn't going to be any problem. The RFMS library was generated using 9 different glycoproteins from several different biological sources. We use several monoclonal antibodies like etanercept, Infliximab, trastuzumab, and EPO, the NIST monoclonal antibody, pooled human IgG, mouse IgG, bovine fetuin for a source of highly branched and sialylated glycans, and yeast invertase as a source for high mannose species. This library contains GU values for 177 glycans that we've tailored to the biopharmaceutical industry. And we realize this isn't an exhaustive library, and some users may have some unique glycans that are in the library. But UNIFI allows users to create their own custom libraries that they can co-search with the RFMS Glycan GU Scientific Library. The library does contain all the major types of N-link glycans. It has high mannose glycans, complex glycans, and hybrid glycans. There's a wide variety of sialylated glycans, including those that have N-glycolyl acid and N-acetyl acid. There's also several glycans that have alpha-linked galactose pairs. And the library also contains GU values for many isomeric glycans, including positional isomers and linkage isomers. So, for example, for several glycans, we can distinguish alpha-2,3 linked sialic acids from alpha-2,6 linked sialic acids. The development of the RFMS Glycan GU Scientific Library was a joint project between Waters and Pauline Rudd and Mark Hilliard of the National Institute for Bioprocessing Research and Training, or NIBRT in Ireland. And they're the ones that really did an exhaustive characterization of the glycans using a battery of exoglycosidases for each glycan sample. And using the exoglycosidases, they were able to selectively remove specific monosaccharides, and this gives us a very precise structural characterization. We get a lot of information out of this. And this characterization is shown in the upper left-hand side of this slide. So for example, they would start off by running the undigested sample, and then they would first remove alpha 2,3 linked sialic acids, and they could look at the shift in retention time and GU values, and then they would remove the alpha 2,6 linked sialic acids, and then they would remove subsequent monosaccharides. So really doing a very thorough job of really pinning down what the exact structure of each glycan is. And then each glycan has also been subjected to mass conformation. And for a lot of glycans, I've also collected MS data. So I'm very confident that all of the GU values and for the structures, they are in fact correct. And why did we go through all of this effort of exoglycosidase digestions and getting accurate mass data? And it was really so that the users didn't have to do this. That all this work has already been done for you, so you can just run your samples, do the library search, and get your results. And I'd like to point out that each sample was analyzed at least 10 times with the mass confirmation, so we do have great confidence in the accuracy of the GU values. Since we are searching a library based on calibrated retention times, we do have a set of conditions for the separation that we do need to follow fairly closely to get the best library search results. We do need to use the 2.1 by 150 millimeter acuity UPLC glycan BEH amide column that has the 1.7 micron particles with a pore size of 130 angstroms. This is the same column that's been used for the 2AB library. That part is the same. The mobile phases, mobile phase A is 50 millimolar ammonium formate at pH 4.5, and mobile phase B is pure acetonitrile with no modifier. We conduct the separations for the RFMS experiments at 60 degrees C. It's a bit higher than what we use for the 2AB library, and we use a gradient from 75% B to 54% B over 35 minutes, or a slope of 0.6% per minute. And this gradient is a little bit different than the one we used for the 2AB library. But the column is the same. The mobile phases are the same, just a couple of slight changes in the conditions from going from the 2AB experiments to the RapiFluor-MS experiments.
We use glucose units instead of raw retention times. We found GU values to be a little bit more stable across different instrument systems or across different columns or even across different days. Raw retention times do tend to shift around a little bit, and so if we search a library just based on retention times, our search results may not be as well as we would like them to be, so we recommend using GU values. To be able to convert retention times into glucose units, we first run an analysis using dextrin that's been labeled with RapiFluor-MS, and we can see this in the figure on the upper right-hand part of the slide. We then tell the software the exact retention time for the structures. For example, we're telling the software that GU4 has a retention time of 7.53 minutes, GU5 is 10.66 minutes, and on and on and on. We can then convert those GU values and retention times into a calibration curve shown just below the FLR chromatogram, and then we can generate an equation based on this data that we can then use to convert the retention times of our unknown samples into GU values, which we can then use to search the scientific library.
In order to obtain the best GU values and get the most accurate scientific library search results for RFMS, we do recommend using our RapiFluor-MS dextrin calibration ladder. It took a little bit of chemistry to be able to get RFMS attached to it, but we were able to do that through an ethanol-amino-urea linkage. And our dextrin ladder covers GU values from 2 out to 30, allowing us to get accurate GU values from very small glycans out to very large ones. So, some of these huge glycans that are, highly branched with, tetra and ternary, 4 sialic acids, a couple of fucoses, maybe throw in some lactosamine extensions. We can get accurate GU values for all of those types of glycans. And to get the best library search results, we recommend running the calibration ladder with each analysis set. And now let's take a look at some fully processed results that we get from UNIFI. And this slide is a recreation of what we would see. On the left-hand side, we see the FLR trace that's been annotated automatically with the glycans from the library search result. And if we click on any one of those structures, we can pull out the extracted ion chromatogram. And here we see 2 peaks when we do that, so right away I know that we're dealing with isomers. And if we want, we can pull out the process mass spectra as well. On the right-hand side of the slide, we see the library search results, and I'd like to explain how the library search works. We do search within a GU window. We recommend plus or minus 0.2 GU units. And after UNIFI has calculated the GU value for a given peak in the FLR chromatogram, it'll search the library for all of the glycans that fall within that window. So for the highlighted peak in the FLR trace, UNIFI determined it had a GU value of 8.18. So the library search pulled out all of the glycans that had GU values between 7.98 and 8.38. Then the software goes back and will determine which glycan out of that list is actually the correct one. And this is where the mass spec data comes into play. It'll go back into the mass spec data. It'll look for an ion, and it can search different charge states. That's not an issue. But it'll look for an ion corresponding to that particular glycan in the mass spectra. If it sees that ion with an acceptable mass accuracy, about 2 ppm, then we say that that glycan is mass confirmed. And if we look at the results for this particular search, the glycan that's highlighted had a delta GU of 0.011 GU units and a delta M over Z of 0.005. So these are both very accurate results. And so there's absolutely no question that the library has given us the correct glycan for that particular GU value.
And now let's take a look at how we can use this workflow in a real-world study where we are comparing Innovator and Biosimilar Infliximab and looking at their glycan profiles. Infliximab has been on the market for about 20 years now. It's an anti-TNF alpha monoclonal antibody. It was originally approved to treat rheumatoid arthritis, and it's now been approved to treat Crohn's disease and a number of other diseases as well. There's been a biosimilar version known as Inflectra that's been developed by a company based out of Korea. The biosimilar version has been approved for use in Europe, Canada, Australia, and South Korea. And importantly, this is the first biosimilar monoclonal antibody that's been approved for use in the United States. And so we thought this would be a very timely comparison to do between the biosimilar and the Innovator Infliximab. To begin our comparison of Innovator and Biosimilar Infliximab, we first looked at the FLR profiles. We were able to obtain 3 samples of the Innovator molecule and 1 sample of the biosimilar. But if we look at the FLR traces, we can see how similar these 4 traces are. There are some slight differences with some of the lower abundance glycans, it appears, but for the most part, the major structures appear to be very similar. We were able to identify 23 unique glycans on the Innovator molecule and 21 on the biosimilar. All of the glycans that were on the biosimilar were also on the Innovator molecule. So there are 2 unique glycans on the Innovator monoclonal antibody. Once we've done the library search and we've determined what the carbohydrates on the sample are, we can start to group those into various classes based on their structural features. And this is what this slide is showing. And we're just comparing different classes of glycans between the Innovator molecules, which are shown in blue, and the biosimilar, which is shown in red. So we're able to classify in this example sugars that are neutral, monosialylated, non-fucosylated, fucosylated, monoantennary, biantennary, high mannose, and those that have alpha-linked galactose units. And I think overall we can see that the different classes are very similar. There appear to be some slight differences. For example, it looks like the neutral glycans are slightly more elevated in the Innovator sample, and perhaps the monosialylated are slightly more abundant in the biosimilar sample. But overall, we think that the different classes are present at a very, very similar abundance levels on both types of molecule. To drill down into the data and really look at specific glycans that are different in their abundance levels between the Innovator and the biosimilar molecule, we can start to look at these mirror image plots. And in this plot, I'm looking at the biosimilar shown on top in green and the Innovator shown on the bottom in black. And the difference in the raw intensity of these peaks is shown as the red trace in the middle. Now, we are looking at raw data in this example, but we believe that since we start with the same amount of starting material at 15 micrograms of each monoclonal antibody, and we inject the same amount, we've already taken care of the normalization, so we believe that this is a valid comparison. Just a couple of things. If we look at this peak at retention time of 12.94 minutes, we see the red trace dips down, then jumps back up. This is just indicating that there are no differences in the peak intensity for that particular for that particular carbohydrate. If we zoom in on some of the carbohydrates that are lower in their abundance levels that are later, around 18 minutes to about 24 minutes, we can start to see that there are some differences in those abundance levels. The interesting thing is that these are all terminated with N-glycosylated acid, and they all appear to be more elevated in their abundance levels on the biosimilar monoclonal antibody. Although it is worth noting that these are present at very low abundance levels. When we start to look at all of the Innovator monoclonal antibodies, we can start to see those patterns that we saw on the mirror image plots starting to really be emphasized for the glycans that are capped with N-glycosylated acid. And overall, it looks like that those types of glycans are more abundant on the biosimilar molecule, and we have 4 examples of those. In general, it looks like these sialylated species were about 3 times more abundant on the biosimilar molecule as on the Innovator molecule. If we look at glycans that have alpha-linked galactose pairs, those tend to be more prevalent on the Innovator molecule. In fact, this hybrid glycan shown on the bottom right-hand corner was only observed on the Innovator molecule. An interesting glycan was the one that had both N-acetylneuraminic acid and alpha-linked galactose on it. What I find interesting about this was it was more abundant on the biosimilar molecule, although it wasn't as abundant as some of the other carbohydrates that didn't have the alpha galactose linked to it. So I think it's really interesting when we look at these glycans that can have some immunogenic responses, that there seems to be a preferential expression for the type of MAB. Again, the glycans that have N-glycosylated acid tend to be more abundant on the biosimilar. The glycans that have alpha-linked galactose pairs appear to be more abundant on the Innovator molecule. It's also important to take a look at the levels of fucose, and again, we can start to look at those levels once we've done the library search. Overall, the levels of fucose were quite similar on both molecules. There was one particular glycan that we call FA26 G41 that appeared to be more abundant on the Innovator monoclonal antibody. But that increase was kind of compensated for by the F6A2 glycan. So overall, the levels of fucosylation on both types of MAB were very similar. To conclude today's talk, I'd like to highlight some of the key points that we discussed. We're very excited about RapiFluor-MS. This is the first molecule on the market that can rapidly or instantly label glycosylamines that can enhance both the FLR and the mass spec signal. Again, this reaction is done in a matter of seconds, but we recommend letting it go for a few minutes. We can then integrate this into a simple sample preparation workflow. We've been able to cut the time investment down from several hours to a few days down to just a few minutes. So we can get the sample preparation done in as little as 30 minutes, but depending on the number of samples, it may take up to an hour. We can then incorporate this simplified sample preparation scheme into a simplified data acquisition and analysis within UNIFI. We're very excited about being able to simplify the data analysis using our scientific libraries to search glycan values. Traditionally, this has been an area that's been very difficult and can be very time consuming. But with the ability to search a scientific library, it greatly simplifies this task, and it makes it more straightforward and a lot faster. We can then take this overall workflow of RapiFluor-MS labeling with our automated data acquisition and library searching, and we can start to develop some very interesting applications. And in the example that I showed today, we can start to compare monoclonal antibodies from Innovator and Biosimilar companies, and we're able to look at what types of glycans are different and what types of glycans are the same. And so with that, there are several people that I'd like to thank. I would like to thank Pauline Rudd and Mark Hilliard of NIBRD. They've been instrumental in assisting with the co-development of the RFMS Glycan GU Scientific Library. I'd also like to thank Matthew Lauber, Ying-Ching Chu, Rose Lawler, Scott Berger, Min Du, Jennifer Fournier, and Alexia Armstrong at Waters for helping put this presentation together. I'd also like to take this opportunity to thank Jeff for giving us this opportunity to present some of the research that we're doing up at Waters in the area of glycan analysis. We're very excited about some of these developments that we believe will really streamline the task of glycan analysis.
Thanks, Bill. That was a great presentation. You provided our audience with some great information about the advantages of the RapiFluor-MS Kit and how it will streamline their glycan analysis workflows. So we thank you for that. Before we get started on the Q&A session, I want to remind everyone once again that this is your final chance to submit questions for our speaker. So hurry up and submit them now. Just type them into the box to the left-hand side of your screen and hit submit. All right, we have some really great questions that have already started to roll in. So why don't we get to answering as many of them as we can. Bear with us for a few moments while we get everything situated on our end, and we'll begin the Q&A. All right, everyone, thanks for joining us for the Q&A. I have a few questions that have rolled in already, so let's get to them. Bill? First question is, can we add glycans to the library? In addition to the more glycans, I have some very unique structures, and I want to be sure that I can use the library as effectively as possible.
Yeah, that's a great question. Unfortunately, we can't edit glycans that are in the library. We've got that locked down. But what we can do within UNIFI is we can create our own custom library. So for the customers that are dealing with some unique structures that they've engineered for their particular protein, we can go in, we can create that glycan structure, we can calculate the data, and we can create a unique library, put that glycan in that library, and then we can co-search with the RFMS GU Glycan library, and we'll be able to get the structure identified that way. But we can't edit the actual library itself. And co-searching a library works just fine. It works great.
All right. So our next question, one of our audience members would like to know, can we use the library outside of UNIFI? I'm using Power for glycan analysis and would like to be able to use this library.
Yes, you can. This was co-developed with NIBRD, and this has all been uploaded into their GlycoBase database. So you can get this exact same data through GlycoBase. You have to register for an account with that. But you can search the data. It is manual. It's not automated like what we would do in UNIFI, but you do have access to that data outside of UNIFI, yes.
Okay, great. Another question, an audience member starts off by saying, I'm working with some very large glycans that elute very late in the LC-MS analysis. How much flex is there with the UPLC condition?
We do have a little bit of flexibility, particularly with the length of the gradient. So we run a gradient from 75% acetonitrile to 54% acetonitrile. if you need to drop that down to say 45% or 40%, that's fine. That's not going to affect anything. You can lengthen the gradient, but we do need to keep the gradient at the 0.6% per minute. What we can't do is change the temperature. If we start to change the temperature, then we start to change the selectivity of the column, and then our library searches aren't as accurate as we'd like them to be. But lengthening the gradient, not a problem. If you want to shorten it a little bit, that's not going to be a problem either. Just make sure that we keep the slope at 0.6% per minute, and we'll get very accurate library searches.
Okay, thank you, Bill. Another question, will the deglycosylation step eventually give a monosaccharide, or was it a mixture of mono and oligosaccharides?
Yeah, the deglycosylation step, we're just using the rapid PNGs-F from New England Biolabs, and it acts as normal PNGs-F does, and it will release intact N-linked glycans as glycosylamines. Yeah, we don't release those as monosaccharides.
All right, thank you. We have one person who, so go ahead, Bill.
And I just wanted to mention the one thing that we have done is we have optimized the buffer with the rapid PNGs-F so that it does work better with RapiFluor-MS. So the buffer is a little bit different. That's why we're co-marketing with New England Biolabs.
All right, thank you. So one of our audience, I guess, missed a little bit of information and would like to know if you could repeat how do you maintain the glycosylamine during PNGs-F treatment, and what keeps it from converting to reducing it?
That's a greite question. The ability to do that, the deglycosylation, is we do use quite a bit high concentration of the PNGs-F, and we do the reaction in 5 minutes. And the half-life of the glycosylamine is a couple of hours. So by getting the digestion done in 5 minutes and then doing all the subsequent sample derivatizations, in another 5 minutes or so, we're able to trap the glycans as glycosylamines and get them reacted efficiently before they have a chance to be converted back down to the hydroxyl. So really, we're just taking advantage of the rapid kinetics and being able to rapidly label things. So we're just taking advantage of the time.
Okay, and it looks like we have our last question. Audience member would like to know, how do you determine the stereochemistry of glycosidic linkages? Is it based solely on the glycan library?
Yes, so this is all ties back into the very rigorous characterization with the exoglycosidase digestions that our coworkers and collaborators at NIBRT did, where they're using very specific exoglycosidases to be able to, very selectively remove amino acids based on, alpha-beta linkages and, alpha-2,3 versus alpha-2,6 linked sialic acids and, the galactose linkages as well, and on and on and on and on down for each monosaccharide. So we're getting that type of information from our very rigorous characterization using very specific exoglycosidases.
All righty, well, thank you, Bill. And with that, it looks like we've come to the end of our webinar. So I want to remind everyone that our webinar will be archived for up to a year on our website at www.genengnews.com. So if you missed parts of it, you can watch it again, or feel free to forward the link to your friends and colleagues, which we always recommend. I'd like to thank Bill again for his informative presentation, and I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks to Waters for sponsoring this webinar. So hopefully we'll see you again at another Gen webinar in the near future. Goodbye for now.